Testing, testing, testing.
Nothing?
Yeah,
Okay.
there you go.
Hello. Okay, so the title of my talk is Ramachandran's Mountain Pass, Exploring Impossibilities. But before I dive into it, I want to start with a question. How does a residue get from one side of the Ramachandran plot to the other? That's what I'm going to answer today. Okay, so our story begins in 1963 when Ramachandran and his graduate student, Saksaran, seen here in the only picture in the entire internet of him, used a simple ball and stick model to predict disallowed, as they called it, or impossible regions of phi-size space. So essentially, for example, this entire central region where phi is close to 0 is disallowed because of an oxygen to carbon clash. Each of these regions can be thought of actually in terms of the clash that disallows them. In these two highest peaks of disallowed energy space, the clash is so bad that it's practically covalent bond. So you can think of all of these regions in terms of what disallows them, or in the case of these two highest peaks, you can think of them as I do as Mount Ramachandran and Mount Saksaran. But back to our original question of why do we care, what's the point? So again, how are we getting a residue from, say, the alpha L region to the extended conformation? Which, if we recognize that these conformations are populated all the time by residues sampling different conformations as proteins are folding, they have to be transitioning somehow. What path are they taking? But this isn't just something that we think is happening during protein folding as residues sample different conformations. There's actually recent examples of proteins that use this barrier to create a conformational switch that controls the activity of the protein. In this case, a single tyrosine controlling ligand binding, or in this case, a tyrosine that swings a loop into a different direction and forms the active site where the catalytic activity is present. And you can clearly see that the residue is in fact going from the alpha L to the extended conformation. Interestingly, these authors in their study calculated energetics for different phi-psi positions, and when they looked at these energetics, they said, oh, we definitely don't think it's going this way. It's definitely going this way. And so they used that to calculate their energetics for this energy barrier. And we'll see why that maybe wasn't the best choice. Okay, so what we can see when we look at this oxygen to carbon distance that describes the central region of the Ramachandran plot, this disallowed region, when we look at this distance, we can see that this actually resembles a classical transition state or a diagram that we're all familiar with from freshman chemistry. Where at the peak, at phi equals 0, this distance is 2.32 angstroms, highly unfavorable clash that completely occludes this region. But as we get to that region, at about phi equals 53-ish, it reaches 2.8 angstroms. Ramachandran's predicted normal approach limit for oxygen and carbon. And then it very quickly goes past 2.7 angstroms, the extreme approach limit, which he predicted in 1963. So that's if you build these things with ideal geometry. So here I am standing up here in front of you talking about impossible conformations and residues that may or may not exist. And that's interesting, it's kind of cool, but what's really cool is these things really exist. They really exist in this impossible conformation. So you pull up all the structures using the protein geometry database that are well resolved, they're at 1.5 angstrom resolution or greater, and they have less than 90% sequence identity. And what you see is that there are these two distinct paths or mountain passes that have residues in this impossible conformation. And if you notice, where phi equals 0, which is our peak transition state, you see it's about size minus 90 and plus 90, which in a 2013 study that calculated energetics using minimization, you see that corresponds to these sort of low energy regions in between here, in between these two mountains, these two mountain paths, if you will. So, are they real, is the next obvious question. I'm sure many of you are immediately doubting the existence of these things. I know I was. So in order to do that, I grabbed all of the structures between phi minus 35 and plus 35, because that restricted me to a reasonable number that one human being could reasonably expect to look at within their lifetime. And I began doing just that. So I pulled up the density for all of these structures, and I went through, and I got things like, for example, this 1 angstrom structure, where the density doesn't really tell you very much, to say this politely. Is this confirmation really there? I don't know. I'm not going to comment on it. And then on the opposite end of the spectrum, you get this 1 angstrom structure where the density is so clear and unambiguous that it makes you insecure about your own solid structures. So going through this process, I was able to discard 33 structures for various reasons, mostly because I was ashamed of them. And then I was able to add 46 structures due to non-crystallographic symmetry, bringing my total up to 146 of these between 5 minus 35 and plus 35. And so the next question that suggested itself to me was, how is this even possible? And if we look at them individually, it becomes apparent that in fact this clash that's supposed to make this impossible just isn't there. The distance here is 2.88 angstroms, colored green here because green is obviously good and red is bad. But what you can see is that this distance is expanded because these angles have actually expanded well beyond the ideal values that you expect, as well as these angles compressing. So individually, you can see that these angles are expanding, the distance is maintaining a satisfactory distance. But that's individually. Are there any systematic trends that we can observe that allow transit through this mountain pass? So to begin, it seems that there's these 2 mountain passes that go through this high energy barrier. But are these 2 mountain passes actually any different from each other? Well, if you take the bottom half of the plot and using really advanced Photoshop technique, you can actually flip it over and mirror it, and then overlay it on the other half of the plot. And what you see is not only do the predicted energetics contours map very nicely, but also all these residues line up on the same path. And in fact, one line describes both of these mountain passes, suggesting that this is just one phenomenon that allows transit through this high energy region. There is one mountain pass. So when we go back to look at the oxygen to carbon distance for these residues as they go through this transition, what we see is these black lines are the ideal geometry predicted structures and distances, and very quickly they go well off the screen because I've just given up on them. But you can see the blue triangles here which represent the real structures, and these purple dots which represent the binned means for these distances. And what you see is they begin to deviate from ideality at about 2.8 angstroms, which is the normal approach limit, and then they peak at about 2.7, the extreme approach limit, and they flatten out, and then they go back to normal. So that's great. I mean, how are they doing that though? So we look at the bond angles, the interior bond angles here, especially these 3 that control this distance, and we say, are they just randomly fluctuating? I think not. How are they expanding, and is it systematic? So we take the same bins, and we get the averages for these bond angles, and what we see is that outside of this unfavorable region, they do match the predicted ideal values. But as you get into the region, they begin to expand and expand and expand until they reach their peak at phi equals 0, which is the exact transition state peak that we expect based on the theory. And in fact, these angles also similarly compress slightly in order to accommodate some of this change. So these green lines here are cosine equations that I fit, and I think I fit them reasonably well. And with those cosine equations in hand, I could regenerate peptides in 1 degree intervals throughout this transition. And instead of saying, take these ideal angles and generate me a bunch of peptides, I could say, take this equation, and given the phi value for that peptide, generate me that peptide. And after doing that, what I observe is that the oxygen to carbon distance actually matches the real structures quite nicely, going down to a minimum at phi equals 0 of 2.66 angstroms, which does respect Ramachandran's early predicted distance of 2.7 angstroms, which I think, hey, kudos to him. But this isn't really the end of the story. The next question that I had was kind of bugging me. One way that people commonly view these transition states, especially when you can't view transition states really, is simulation. And so if I simulated these things, wouldn't that be just as good? And the answer is, kind of. So I used Amber 12 with the F99 force field, and I simulated these residues by restraining phi and psi. If I gave them either my ideal geometry with the new cosine equations, or I gave it the old ideal geometry, either way, with either starting point, it came to the same solution for every individual position. And a couple of the problems here are that it doesn't let anything get closer than 2.8 angstroms. And it does deform the angles systematically, but it doesn't do it in a way that matches what we observe in the natural structures. So for modeling and for simulation, we need to incorporate a little bit of new information about what's allowed and what's disallowed, or what's impossible, or maybe not so impossible. In conclusion, native structures have trapped these high-energy transition state residues and allowed us to directly observe them and to generate models that describe this transition state. And we can see here the new one, proud in solid colors, and then the ghostly dead old idea here, which is a little bit pinched together. It doesn't look that dramatic in this picture. And finally, this thing's not very loud. I'd like to thank my mentor, Andy Karplus, for putting me on this project and giving me a chance to just do a bunch of crazy stuff and just throw a lot of things at the wall. I'd like to thank Sean O'Neill because he taught me a lot of programming, especially R, which I mostly use for this, and I didn't know any programming, and I'm embarrassed about all my early programming. I'd like to thank Dale for putting up with my 1,000 questions, only like 100 of which actually had answers. And I'd especially like to thank Arden, Camden, and Kelsey for putting up with my general shenanigans in the lab. Thank you, and I'll take any questions.
So Ron Ray's group has done a lot of work on this n-pi star interaction, and if I remember correctly, it looks a lot like some of the figures that you've shown. So can you comment on whether or not there's a relationship there?
I honestly don't even know what the n-pi star interaction is. I'm sorry.
Any more questions?
So I saw in one of your previous slides that one of the main compromises by the kind of amplitude of your cosine graph was in the C alpha NC angle, but did you also look at the omega torsion angles and kind of deformation around the peptide bond?
Yes, so I did look at the omega angle. I don't actually have the figure for it with me, but it's not that interesting. It's not that compelling. The first time I looked at that graph, I thought, oh my god, there's such a big change, but actually there was just a cis peptide in my data set. But after that, there's kind of like a range from like plus 5 degrees to minus 5 degrees, but it's not systematic. It seems to be just something that individual residues go through as one of their ways to relieve some of the strain.
So any more questions? Let's thank you, Andrew, for